All right, Science 30. So the next video here is on mutations. When we talk about mutations, these are changes in DNA. And sometimes mutations are bad, of course, but what we don't realize is sometimes mutations are actually really, really helpful. And so a mutation itself isn't a bad thing necessarily. It could be a wonderful thing, and it could be really beneficial to a species, and it could cause a huge amount of evolution later. But a mutation, nonetheless, is a mistake. It isn't something that's supposed to normally happen in that organism's genome, but something has caused the mutation to occur, whether it was radiation or chemicals or just honestly a mishap in crossing over and just something went wrong, something was deleted, something was omitted, something was added, something was substituted, but something changed which wasn't supposed to, and it modified what was produced. The cell can usually, and the body can usually genetically fix those mistakes. You have a bit of an autocorrect system going on in the genome, and it's able to see spelling errors in your genetic code. When a mutation occurs, it's able to usually fix it or try to do something to make the results of that mutation not catastrophic. However, you can't always uh, catch those, right? The body doesn't always catch those mistakes. So just like you could, proofread an essay over and over and over again, or you could proofread a text message that you've read over and over and over again. You've had your friends you screenshotted the text message, had your friends look it over, make sure you said everything right, and then you hit send on it to the person you're trying to send it to, and then you look it over one last time and you realize, ah, crap, I actually did make a mistake, and I spelt this one word wrong and nobody caught it, right? And you feel dumb. But it's the same thing in the body. You don't always catch those mutations, and sometimes it just changes the functioning of certain things, and other times it might stop something from working altogether. So mutations are either good or bad depending on what happens in that context. But importantly, mutations are um, able to be passed on. And so, because mutations are genetic, if there's a mutation that's beneficial, it can get passed on, assuming that organism survives long enough to reproduce. If it does not end up being beneficial, then if that organism still lives long enough to be reproduced, it might pass that mutation on, and that mutation might end up actually being harmful to the organism. So, beneficial mutations are obviously things that give an organism an advantage to survive. If it makes them more fit for their environment, then they can survive longer and reproduce more. So mutations are either chromosomal, or we can call them a different type of mutation, a point mutation. So chromosomal mutations are large changes in the actual chromosome. So an entire chromosome might be missing or added, and chromosomes contain lots and lots and lots of different genes that code for lots and lots of different things in the body. So chromosomal mutations are usually quite severe and quite catastrophic. Typically, it's chromosomal mutations that are going to end up being detrimental and negative. Point mutations, on the other hand, can sometimes not matter at all. Sometimes nothing changes. Sometimes only something is slightly modified. Sometimes things are catastrophic, and other times they're actually quite helpful. And so point mutations are much smaller changes. This is an addition or a deletion or a substitution of like one or two or small numbers of base pairs. So like those A's, those T's, those C's, those G's. Something's changed, and so it's not pulling the right amino acid in to make the protein or the hormone or whatever is trying to be produced from that genetic code. So these are much smaller and more specific mutations than chromosomal mutations. So chromosomal mutations can be diagnosed by doing something called a karyotype, which is basically a diagram that looks just like this. Here's all 23 of your chromosome pairs laid out. It's a karyotype. And so we like to look at genes and chromosomes by looking at their size, their banding pattern, which is the bands or the different genes that are on there, and the centromere position. The centromere position is the point where they attach. So this diagram here shows it a little better, but you see how the X's are sort of attached at that sort of center point? Some of them are more visible than others here, but that's called the centromere, the sort of sticky part, the junction where the two copies of each chromosome are connected. Okay, so we can detect genetic disorders using two methods. One method is called amniocentesis, which uses a needle and it pokes it right into the baby's embryonic fluid, and we can check if baby's genes are okay using that embryonic fluid. Or we can use something called CVS, chorionic villus sampling. And this is where you take a sample of the chorion, which is a portion of the fetal placenta, And it is also going to provide you with information about how baby's genetics are doing early in the pregnancy. So you could detect if there's chromosomal disorders that might affect quality of life for the child. Here's just different chromosomes here. So this is trisomy 21, for example. You have 3 copies of the 21st chromosome. Remember, you should only have 2 of each. This is called Down syndrome. Turner syndrome, you're missing a copy of your X chromosome. And so you should have at least 2 X's if you're a female, or 1 X and 1 Y. And in this case, if you have Turner's, you only have an X and nothing else. You don't have another X and you don't have another Y. Jacob syndrome is the opposite. You have an extra Y. So you have an X and you have a Y, and then you have a third Y, or a second Y. So you have 3 sex chromosomes where you should only have 2. So that's not right. So it's a chromosomal mutation. Klinefelters is an example of one where you might have 2 X's and 1 Y, right? The opposite of Turner's. The opposite of Jacob's. Jacob's is 1 X and 2 Y's, and Klinefelter's is 2 X's and 1 Y. These individuals, they live, but they just have different sort of characteristics in their bodies. They're going to have, because of that Y chromosome, they're going to have male characteristics, but they also have female characteristics. Point mutations, so addition, deletion, or substitution, are those much smaller changes or mutations, and this is something that can either have a catastrophic change or a minor change or no change at all, even if the mutation occurs. And the big thing here is you're adding, deleting, or substituting a letter or a couple of letters. And so it'll change, could possibly change the protein that's made because the amino acid that's produced is different. So it's like, I mean, you could think of it like, imagine like you're teaching and then halfway through the year you get a substitute teacher. It's a different teacher and they're teaching the same thing, but they do it a little differently. So that's like substitution, and versus adding or deleting are like actually adding a letter into your genome or removing a letter completely from your genome. Deletion and addition mutations can be more dangerous than substitution mutations because addition or deletion will actually completely stagger the codons that are read by the amino acids during protein synthesis. So if you'll notice here, TCG, ATA, if you add an A, you get TCA, and then everything after that is staggered by 1 base pair, which means that everything else is probably going to be read wrong and pull the wrong amino acid in, which is significantly more detrimental than a substitution would be, where you just change 1 letter, C to T, and everything else stays the same. So addition and deletion mutations are more severe than substitution. Okay, so here's a little practice. I'll pause it here. You can check to see if it's substitution, deletion, or addition. Okay, here's the answers. So only 1 nitrogen base was changed in this one, so it's substitution. Here, lots was changed, and so this must have been deletion. We staggered backwards. So again, point mutations. If you have a substitution reaction, only 1 amino acid is changed. If you have a deletion or addition, many amino acids are changed from the original. And so here only leucine was changed out, but arginine and valine were still made. In this deletion example, you no longer have methionine, arginine, and valine. You now had everything because you removed 1 base pair. You now had cysteine, glutamate, and phenylalanine produced. And then even with addition, you had 3 totally different amino acids brought in again. And so with addition and deletion mutations, significant differences can occur, which can be very detrimental, as opposed to a substitution mutation, which really isn't going to change a whole lot necessarily. Some more examples here for you. Regarding antibiotics, I will do this very briefly in the next video. Okay, thanks guys.